So we were discussing polysaccharide, and that just we want to see the main four structure. We just saw like starch, cellulose, glycogen, chitin, these all. Let's start with the starch. What exactly is this? So starch is like, if you talk about the monomer of a starch would be what? Monomer of starch is alpha glucose. Alpha glucose. So if you don't know what these both like alpha and beta glucose are there, if you don't know, let me just tell you. When you find a bond, like if you made a chain and H is present on the above side and OH is present on the downward side, OH downward side, H on the above side, so that chain is known as alpha glucose. When we talk about OH is present on the above side and H would be present in the downward part, that would be known as beta glucose. Beta glucose. Okay, so when we hit, so starch is monomer of alpha glucose, and you'll find that starch, like we know that whatever we eat we get starch. Starch in mouth is converted into what? That is converted into maltose. Maltose is the malt sugar, maltose, and then it will be converted into glucose, and that glucose is directly taken by our body, because we always need glucose no matter what exactly process this have done. Okay, then when we talk about starch, you'll find two more structure in this. What two more structure you find? First one is amylose. Second one is amylopectin. Amylopectin. You'll find these are unbranched structure, unbranched, whereas amylopectin, which is branched. If these are not branched, that means we can say these are linear structure. So this structure would be linear. That means if it's not branched, this would be straight. This would be like carbon attached with these all simple. Okay. Then in this you'll find 50 to, like in the chains you'll find 15 to 20% of this amylose. 20% amylose, whereas 80 to 85% would be amylopectin. 80 to 85. Because we know unbranched structure, not unbranched but branched structure is a little more. Okay, so that's why. Then when we talk about the primary source of starch, primary source of starch is a lot of like cereals, grain, whatever you eat, cereals, grain. This is basic, no matter if it's present in, if I'm writing that in amylose or amylopectin. Then when we talk about uh, what exactly it will produce or which color would be present when we put this in a test tube, not test tube, but when we talk about iodine test, we know in iodine test you'll find two colors. First is not two, but you'll find many. But if we talk about the iodine test of starch, then you'll find which color? You'll find blue color. So this is very important MCQs from a book as well, from a past paper as well, that when we put starch into iodine test, which color you'll find, and that color would be blue. Okay, and in glycogen and other sources you'll find red. In glycogen you'll find red. So that's a different difference between these both. Okay, then